Okay, welcome again everyone to Biology 2. So this is the second module of Biology 2, and after finishing about Mendelian inheritance, we will now proceed with non-Mendelian inheritance. So this would be the extension of Mendelian inheritance, and we'll be talking about this very important topic. Now, before we begin, take note that the objectives for module number 2 is number 1, to describe the dominance and differentiate it from codominance and incomplete dominance. And number two, to predict the expected phenotypic and genotypic ratios in the filial generations of a monohybrid cross with complete, incomplete, and dominance and codominance. And finally, the third objective for today's discussion is perform a seat work on determining phenotypic, genotypic ratios, and patterns of inheritance on the various degrees of dominance. So with all of those objectives for Module 2, we can now proceed with our discussion. And first of all, let's just go back to what we have been discussing for the past few meetings. So you will be noticing something very important here. These are mental space, or the characteristic of mental space, which he made use in his experiments. And take note of the following: for the seed form, round and wrinkled; for the seed color, yellow and green; for the pod form, inflated, constricted; for the pod color, green and yellow; for the flower color, purple and white; flower position, eggshell and terminal; and stem length, tall and short. And probably you already know what I'm trying to point at, is that you will be noticing that they are always in pairs. So in this case, you will have a dominant, and then you will also have a recessive character. So you only have two forms, or two alternatives for those particular traits. And that's a very, very important characteristic of Mendelian inheritance. So let's look at this one again. So when we say Mendelian genetics or Mendelian inheritance, we are actually looking at complete dominance, and that's the first of which, the first of Mendel's laws or principles. And as we have already mentioned a while ago, there are only two possible phenotypes: either there is the dominant or the recessive. And for this one example, for example, the tall plant is homozygous dominant and the short plant is homozygous recessive. So when you cross them together, the tall and the short, you will be producing a tall plant, which is the phenotype is the phenotype would be the same as that of the dominance. You can only have two possible characteristics when you are talking about Mendelian inheritance, particularly complete dominance. And when we're talking about the important ratios that you have to remember about Mendelian inheritance, first of all, let's talk about the monohybrid cross. So if you have a heterozygote cross with a heterozygote, you will always have a genotypic ratio of 1 is to 2 is to 1, where the 1 is the dominant, homozygous dominant, the 2 would be the heterozygote, and the 1 would be the homozygote recessive. The phenotypic ratio of a heterozygote crossed with a heterozygote will always be 3 is to 1 ratio, where the 3 would be the dominant phenotype. There are 3 of this, and then the 1 would be the recessive phenotype. Going back here, if we have a test cross, and when we say test cross, we have a homozygous recessive that is crossed with an unknown, but for now, it is called a heterozygous test cross because the other parent will be a heterozygote, big A, small a. If that is the case, the genotypic ratio will always be 1 is to 1, and the phenotypic ratio will always be 1 is to 1 as well. And this will be showing us why. Take note of this one. If you have a heterozygous tall and a homozygous recessive dwarf, so the big T, small t, and a small t over here, if you get the gametes, you will get a big T and a small t for the first parent, while the homozygous recessive will always have a small t and a small t as their gametes. When you cross them together, you will produce a big T small t and a small t with the genotypic ratio of 1 is to 1 ratio, because both of the combinations, any combination, will always be resulting to big T small t and a small t and a small t. So the genotypic ratio is 1 is to 1. One small big T small t and one small t small t. If you're gonna be asked about the phenotypes, of course, this will be tall and dwarf. And what's the phenotypic ratio? Since you have only one big T small t, then you have only one tall. There is only one small t, then you have a one dwarf. So the phenotypic ratio will now be 1 is to 1. So again, going back here, in Mendelian inheritance, you have to remember that the genotypes, the ratios that you have to remember is that for monohybrid crosses, a hetero by hetero will always have a genotypic ratio of 1 is to 2 is to 1, a phenotypic ratio of 3 is to 1, And a heterozygous cross, wherein you have the heterozygote cross with a homozygous recessive, will always have a genotypic ratio of 1 is to 1 and a phenotypic ratio of 1 is to 1 as well. Now, if you have here a dihybrid cross, then you will also have this automatic ratio, and a hetero by a hetero for both gene pairs in both of the parents will result to 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1 ratio, genotypic ratio. And the phenotypic ratio would be 9 is to 3 is to 3 is to 1. And we have already discussed that before in the previous videos that we had. Now, if a heterozygous test cross was done for a dihybrid cross, so in this case, you have here a heterozygote for both gene pairs for the parent number 1, and then you will have a second parent, or the second parent would have homozygote recessive for both of the gene pairs. Then the genotypic ratio is 1 is to 1 is to 1 is to 1, and the phenotypic ratio would be 1 is to 1 is to 1 is to 1. But as we continue with our discussion, you will be taking note that there are other kinds of characters that do not follow the Mendelian inheritance, and that's what we're going to be talking about. We're going to be talking about the non-Mendelian. So take note of this one. There are two different types of hair, and the reason that I showed you a while ago the characteristic of this, because it's all alternatives. There are only two forms here: yellow, green, wrinkled, smooth. But take note of these different hair types. You have three phenotypes here. One is straight, the other is curly, and what's in the middle? It is wavy. So we have three phenotypes here, and this does not follow the 3 is to 1 ratio of Mendelian inheritance in terms of what we call as the phenotype. So we proceed with the non-Mendelian, and this is homozygous straight, heterozygous condition, while this is homozygous curly, which we will be talking about a little bit later. Okay, so these are the other types of inheritances that does not follow Mendelian inheritance. So the ratios you have remembered are only for the Mendelian inheritance. So they have different ratios and different genotypic and different phenotypic ratios. And what are these non-Mendelian inheritances? It includes incomplete inheritance, codominance, multiple alleles, polygenic traits, and sex-linked characters or traits. And for this time around, we will be talking about incomplete dominance and also codominance. And for the next meetings, we will have the others to discuss about. Okay, now let's talk about this one. So not all inheritance is based on the rules of complete dominance. So you have this following examples here. So you have here the sickle cell anemia, and then the different colors of the eye. So there's no this it's just not two alternative, but there are several forms and alternatives. You have also here a cat, where you will also have a different kind of trait as well, and also the flowers here. It's not only later we'll be talking about that a little bit later. Okay, so for the degrees of dominance, so today we're gonna talk about we already finished with complete dominance. We already finished that, and then we'll be talking about the first two non-Mendelian, which is incomplete dominance and co-dominance. Again, the reason I discussed about the ratios a while ago, you will be noticing that for the incomplete and codominance, you will find they have different ratios compared with the Mendelian inheritance, which is the complete dominance. Okay, let's start with the incomplete dominance first. So the description of this, the description of this incomplete dominance is the following: neither allele is dominant. So in the complete dominant, we have one that's completely dominant and the other is recessive. And I have already shown you the hand sign, wherein if you have a dominant and a recessive combined together, the dominant will be suppressing the expression of the recessive, so you only see the dominant. But in this case, the one of the allele and the other allele will be both expressed, so you will see both of them to a certain degree combining and expressing themselves. So when an organism is heterozygous for a trait, it will then show a third phenotype. As you can see here, this flower is red, this flower is yellow, and then what will we produce? So the combination of the red and the yellow will produce a third phenotype, which is what we call as an orange colored flower here. So the third phenotype is the blend of the other two, and in this example, you will see that the A gene contains the R, and the A gene here contains the yellow. So when you mix them up together, this is a heterozygote. The red will be produced, the yellow will be produced as well. And then since they do not completely dominate each other, then they will be mixing together, producing what we call as the orange color, at least for this flower. So the R and the Y will be representing the different alleles. So it's quite easy to remember. It will always be producing a third phenotype. Here's another example, and this would be for a red flower and a white flower. So supposing that the red flower is big R, and the white flower is small r. So when you combine them together, as you already know, it will produce a heterozygote, big R, small r, or the heterozygote. The red will be produced, the white will also be produced. They will mix together. So in incomplete dominance, you will produce a pink flower for the F1 generation. So let's talk about the F2. What if you cross a pink flower with another pink flower and F1 with another F1? Then you will have the following Punnett square and progenies produced. So for the big R, small r pink flower, which is F1 generation, you will get the following gametes, which is big R and small r. And you will also have the same thing here, where you will get the big R and the small r gametes. So if you combine the first pane, the big R and the big R, you will get a red flower. The big R and the small r, you get a pink flower. The small r and the big R, you get a bigger, smaller is a pink flower, and the small r and the small r, you get a white flower. So you will be noticing that the genotypic ratio is 1 red, 2 pinks, and also 1 white. And that's the genotypic. The genotypic ratio, I mean, is the one big R, two big R, small r, and one smaller small. So 1 is to 2 is to 1 ratio would be the genotypic ratio. But for the phenotypic ratio, it's quite different because in Mendelian, it's 3 is to 1. But for incomplete dominance, it is 1 is to 2 is to 1 because you have 1 red, you have 2 pinks, and then you have 1 white. So that is a very different phenotypic ratio for incomplete dominance. Okay, here's another example. What is the probability of pink flowers if pink flowers are bred with red flowers? So in this case, if the pink, which is the F1 or the heterozygote, is crossed with a red flower, which is a homozygote, so you will have this. The red will be big R, and the pink will be bigger, smaller. So simply combining them, you have a bigger here. The second pane is a big R. The third pane is a bigger small r. And the last pane is a heterozygote, a big R and a small r. So we already know that this pane would be red. The second pane would be red. The third pane would be a pink because it's a heterozygote. And then the last one is a pink as well. So you will have here 50% chance of pink flowers. So the genotypic ratio is 1 is to 1. 1 big R is to 1 bigger small r, and the genotypic ratio would be 1 red is to 1 pink, which is 50% chance of having pink flowers. So it's quite easy to remember this. Okay, another example is horse coat color, wherein the brown horse given by the big B, when it is crossed with a white horse given by the WW, it will produce a golden palomino or cream coat, which is the B and W over here. So it's a mixture of the brown and a mixture of the white, producing the golden palomino or the cream coat. So it's quite easy to understand it why it is incomplete dominance. Another example here, even the pitch of our voice is incomplete dominance, wherein the lowest pitch is given by the big L, and then the highest pitch is a small l, but the intermediate range baritone or the baritone would be big L, small l. It's a heterozygote. So just for fun, what do you think is the genotype of Darth Vader? Something like that. So we already know that the voice of Darth Vader is very low. So the genotype of Darth Vader would be big L and big L because it's a very low pitch of voice. And then interestingly enough, I searched the internet kasi hindi ko because I don't really know, but I found out that Johnny Depp is considered to be a baritone. He sings as well, and some of the articles said that he sings very well. So if he is a baritone, then he is a big L and a small L. And that's for the incomplete dominance. Let's now